was a bio-penguin. So hopefully that we have gotten this all figured out. There shouldn't be any more issues, I hope. So I'm Mrs. Jones, and I teach AP Bio, and so this is a Unit 5 review where we're going to kind of go over we're going to go over meiosis. We're going to look at heredity. We're going to look at a little bit of pedigrees. So I hope that those that were originally in the other live have moved over and that they have found the review. So I'm sorry for all the tech issues. I thought that we had gotten those all figured out straight away. So in case you don't know, I've got a ton of free resources for you. So if you go to apbiopenguins.weebly.com, there's a 396-page review guide. There's TikTok videos where I've gone through every single topic in the CED. There is FRQ Fridays where I've gone through every single FRQ from 2013 to 2023. You need to be practicing your FRQs. So that's a resource you could use. You can either pause the video as you're going. And by pausing it, you can kind of answer the question on your own and then check the answer, as well as I have exemplars built in that were released by College Board. I'm also posting every single day on my Instagram, well, Monday through Friday in my Instagram, where I'm going to post review questions each day. Thank y'all for coming back, by the way. So I am posting every day on my Instagram. We're currently, I think, on 5.5 is what we did on Friday. So we're going to be on 5.6 tomorrow, and then we're moving into 6. So we are kind of, doing one each day, just to kind of help get a little bit of review. So we are 7 weeks. Tomorrow is 7 weeks from the AP exam. So you might be thinking, like, what kind of plan do I have to help me study? I created this 8-week study plan. You are a little behind, but it's okay. So if you want to use this, we'll have some modifications that you could make to kind of help yourself to review. With us being so far in advance, you do have more time, so you'll be able to spend less time each day on the review. You could probably spend, 15, 20 minutes each day doing a little quick review. And that way you're not like overwhelming yourself trying to study everything. And I do have quizzes, codes open. These codes were made specifically for social media sharing, so they shouldn't hit that 1K, but if they happen to hit the 1K mark of 1,000 people playing them, then if you just go to my website and use that home drop-down screen, you're going to see that there is quizzes where I have actually all 120 games open all the time for y'all. So modifications you can make to this plan. So number one, since we're already over here on the 16th and we're supposed to be starting Unit 2, which you haven't reviewed Unit 1, you can skip these breaks. So every Saturday I have a break set up for you. So you could skip that break and just work straight through. So you would be spending 6 days on each of your units. That will allow you to finish in time for the exam. Another option, Units 4 and 5 are kind of short. So you could combine whatever week Unit 4 and 5 are so that you won't have to try to, catch up. You can just start today on Unit 1. Or you can just shift 1 week. So we just take whatever we have on Unit 1, we just move it down and just shift 1 week down. And then you just don't review Unit 8 because theoretically that is the most recent content that you studied in your class. So you wouldn't really need to have that, review for yourself. And of course it keeps going. So here's April. And then there's May, and don't forget, AP exam is Cinco de Mayo at 8 a.m. Yeah, I know, exciting. So today's plan, I'm going to do my, quick rapid fire review of content, and then we'll get into the submitted Q&A from in advance. So there are some students that submitted questions. Happy to answer questions as you go, but the goal is that we stay on Unit 5 content today. I will do, I have a question box on my Instagram where you can post questions anytime, and I'll answer those on my Instagram. But today we're focusing on Unit 5 and doing quick review, and then we'll do the little Q&A for you. So we'll do meiosis. We're going to talk about genetics. We'll look at, there was a practice question built in from somebody, and then of course that Q&A. So meiosis, last time we talked about mitosis, which was making more of our somatic cells. They were identical cells. They were going to have, they were still diploid, and we went through that one round of replication and one round of division. So meiosis still goes through interphase. We still are going to make the copy of the genetic material, just like we saw in mitosis. But now we're going to be looking at making of the sex cells, the reproductive cells, so your sperm or your egg. And so these cells are going to have to be haploid cells. So we're going to end up going through 2 rounds of division. So you're going to notice here that I have a 1 up here. This is prophase 1. We have the exact same steps that we saw where we go prophase, metaphase, anaphase, telophase. Okay, so the exact same steps, but now they're slightly different. So prophase 1, we still have our chromatin condensing. Our sister chromatids are going to find each other, as well as our homologous chromosomes. So sister chromosomes are identical of each other. So chromosome 1 maternal And then its duplicate, these would be sister chromatids. And then chromosome 1 paternal with its duplicate, those are also sister chromatids. But together they make homologous pairs or homologous chromosomes. So they're going to be the exact same size, but it would be the maternal and the paternal set of them that makes them the homologous. So in prophase 1, we're going to see crossing over. This has to do with when our sister chromatids exchange their genetic information, which I did not get. We'll pretend like these are orange. So what you could have, sorry, I dropped the marker. Okay, so if these were our chromosomes, right? So we have maternal and we have paternal, just pretend that that's orange, okay? What's going to happen is that these two interior chromosomes, okay, these non-sister chromatids, they're going to cross over, and by crossing over, they're going to exchange their genetic information. And so now I'm going to end up with a chromosome that is paternal, another chromosome that's paternal, and they'll have two that are recombinant, okay? So they're a combination. They're going to have some paternal and some maternal and some maternal, some paternal. So they're like combinations of those, okay? So I hope that that showed up with my little camera. Yeah. And so that's when crossing over. So, oh, I made myself a picture. That was smart of me. So here you see that chiasma where they're crossing over and then they exchange that genetic information. So in meiosis 1, we are looking at our homologous chromosomes and we're looking at the division of those homologous chromosomes. So same step, we move past prophase into metaphase. And again, we have that 1 because we're going through our first round of division. So metaphase 1, our homologous chromosomes line up on that metaphase plate. And this is when we have independent assortment. Each of those chromosomes, because we have 23, pairs, are going to independently line up on that metaphase plate. And so this is going to allow for a lot of genetic diversity due to the fact that which direction they're going to line up on to allow for us to get to their opposite sides. Anaphase, our homologous chromosomes separate. Now, notice here that I'm specifically talking about my homologous chromosomes. We need to make sure that we are very clear in this when we write our FRQs, that you are clear that it's homologous chromosomes. Don't use pronouns. It split. They split. No. I need to know it's homologous chromosomes. We're seeing the homologous chromosomes line up on the metaphase plate. Homologous chromosomes are segregating and moving to opposite poles. You need to be very clear in this. And then we go into telophase. Telophase 1 is going to be where our nuclear envelope forms around that new haploid daughter cells. Yes, after meiosis 1, the cell is haploid, okay? It's like we have, as I said, we have our paternal and our maternal, okay? They move to opposite poles. This cell is going to have it's two of the same chromosome, okay? So we are haploid. We only have one set of the genetic information. There may be, duplicates, and there may be two of them, but it is still the same genetic information overall, okay? So then we move into the second round, prophase 2, where we're going to see our chromatin condenses, sister chromatids align. This is the exact same thing we saw with mitosis. Again, metaphase 2, sister chromatids line up on the metaphase plate. In phase 2, sister chromatids are going to separate to opposite poles, and then we form our nuclear envelope around those haploid daughter cells. Okay, so everything we already saw with mitosis, now we're seeing in meiosis 2. Okay, so again, meiosis 1 was looking at the separation of homologous chromosomes. Meiosis 2 was separation of our sister chromatids. Okay, so what is the difference between mitosis and meiosis? Okay, so let's look at it. First thing, let's look at the parent ploidy. Okay, in both of these cells, the parent starts out being diploid. So the parent is going to have two sets of chromosomes, 2N. And then how many rounds of replication it goes through? They both go through only one round of replication. But the difference is how many rounds of division. So mitosis goes through one round of division, while meiosis goes through 2 rounds of division. That's what's going to allow you to have haploid cells and end up with 4 cells in the final product, right? We have 4 cells that we make with our meiosis and only 2 cells that we make with mitosis. And that daughter cell is going to be diploid in mitosis and it's going to be haploid in meiosis, okay? Now, how does it compare to that parent cell? In mitosis, they're genetically identical. These are identical to the parents. Versus in meiosis, they're genetically distinct. Again, I only have one set. But also looking at that, we've had crossing over that allowed for recombination. And we had independent assortment. And that independent assortment allowed for there to be differences. You've got maybe chromosome 1, 3, and 5 are maternal, while chromosome 2, 6, and 8 are maternal. So you've got new combinations that weren't there in the original cell. And then crossing over takes place in meiosis 1, but does not take place in mitosis. And then independent assortment takes place in metaphase 1, but does not take place in mitosis. So you're seeing that we have a slight differences there. Do you need to know about chromosomal mutations? Yes, that will go later on. We'll talk about chromosomal mutations. So in terms of our inheritance patterns, right? So we need to think about, this is where all of those Punnett squares are. You absolutely love Punnett squares. I know you do. So with those Punnett squares, just realized you can't see the screen. With those Punnett squares, we're going to look at complete dominance. Complete dominance has to do with that the homozygous dominant and the heterozygous are going to look identical. They look the same, okay? A dominant allele is going to mask the recessive allele. So it's important that we understand dominance. Dominance doesn't mean prevalent. It doesn't mean normal. Dominant allele just means traditionally that it's making a protein. It's making an enzyme. It makes something, while the recessive is usually going to make, it's not going to make a protein. It's going to make an inactive protein. It's going to make something that's not folded correctly. The dominant trait is traditionally going to be able to mask the recessive because it actually makes something. Examples of things that are dominant that are not prevalent: fingers on your hand, right? Polyploidy, having more than 5 fingers, is actually a dominant trait. But I would say that most individuals have 5 fingers, and we are all homozygous recessive, okay? So don't say, oh, well, the normal trait. No. Dominant just means that it's masking or it's covering up. So with our complete dominance, our homozygous dominant and our heterozygous can look the same because of the fact that dominant allele is going to synthesize enough that they are going to be the exact same. Codominance. So we're going to see that both the dominant alleles will show up. So as you see here, we have blue, we have yellow, and then this individual has both the blue and the yellow genes, and you can see them together, okay? Because they're both dominant, they're both synthesizing the material. And then incomplete dominance is that we're going to see that the two are going to blend. Okay, you'll see a combination of blending of these two dominant traits. So we have blue, we have yellow, and then we have green because we're seeing that blending of it. So they're both making the material and it just kind of blends. Common example you see with codominance would be like blood typing. Incomplete dominance, you usually see like snapdragons and the pink flower versus red and white flowers. And so when we think about these crosses, you'll probably have monohybrid or dihybrid. You might not have anything higher than that. We really don't know what you're going to have. It's not like clearly stated in the CED how many you'll have to work with. But some things you want to keep in mind. Monohybrid means that it's heterozygous for one trait. So you have, capital A, lowercase a. That would be monohybrid. And then a dihybrid would be heterozygous for the 2 traits. So capital A, lowercase a, capital B, lowercase b. So that's 2 hybrids, okay? And then complete dominance, when you do that cross in a monohybrid, it's going to give you a 3-to-1 ratio versus a dihybrid gives you a 9-to-3 to 3-to-1, okay? Now, if we were to look at incomplete dominance or codominance with monohybrid, it's a 1-to-2-to-1 ratio. And then dihybrid with incomplete or codominance, it's a 6-to-3 to 3 to 3 to 2 to 1-to-1. So you see that 1-to-2-to-1 ratio kind of built in, and you just multiplied by 3 to get it to be this. So it's just that it's a 2, 1, 1, 2, 1, 1, and then you multiply those by 3, and that gets you 6, 3, 3. So whenever you do any of these Punnett squares, I always recommend that you do a monohybrid cross. So you do a 2x2 square, and then you'll multiply to get the final numbers. Okay, so if we were looking at, I don't know, yellow and green, and we have a yellow plant. Okay, so this is going to be heterozygous for yellow, and so you can see that we're going to end up with 3/4 of them are yellow and 1/4 of them are green, because 1, 2, 3 are yellow, and then 1/4 right there is green. If we were to do the same thing of round versus wrinkled, I believe, we're going to see that 3/4 of them are round. So again, 1, 2, 3, these 3 are round, and then this one would be wrinkled. We're looking at complete dominance in this example. So if I want to know how many of them are yellow and round, okay, instead of drawing a big 4x4 Punnett square, I find that it's easiest to do 2 of these 2x2s, and I just multiply them. Well, there's 3/4 chance of yellow and 3/4 chance of round. So 3/4 times 3/4 is 9/16. And you can do that for all of them, where you say, okay, well, yellow, round, 3/4 times 3/4 gives you 9/16. You do the yellow and the wrinkled. So 3/4 times 1/4 gives us 3/16. And you can do that for each of them, green and round. And then, of course, green and wrinkled. And that would give us our 1/16, okay? And you can see here we have a 9-to-3 to 3-to-1 ratio. Whenever you're doing any of these problems, a good idea would be to check your math at the end, okay? So the way to check is, does the numerator add up to the denominator? So 9 plus 1 is 10, 3 plus 3 is 6, so 10 plus 6 is 16. So I have accounted for all of my possible options here, okay? So that's a good way you can kind of check yourself at the end. So also, what is the inheritance patterns of it? So something can be autosomal. If it's autosomal, that means it is on one of the autosomes. We have 22 autosomes, and they have one of our sex chromosomes or our allosomes. And so the allele is located on that non-sex chromosome, is on one of your autosomes. Versus a sex-linked trait means it's going to be on our X link. It's usually going to be on an X chromosome. There is Y-linked where it could be on the Y chromosome. Traditionally, they look at it being on the X chromosome. What we'll see is that it's on a sex chromosome, traditionally X. So I always just remember it's X-linked right here. There's an X and sex. And then maternal inheritance means that it's found in the DNA that's in the mitochondria or the chloroplast. As a reminder, mitochondria and chloroplasts, okay, is going to be found in your egg. And so if we're looking at a plant, that's of course going to be looking at the, we have our egg is going to be in our female or our ovule, okay, in the plant. And then if we're thinking about any other animal, usually we're looking at that egg being in the female, okay? And so if we have it in the female, then it's going to get passed on to the offspring automatically because the mitochondria only has a single circular piece of DNA. If you remember, mitochondria and chloroplast, it's believed that they were prokaryotes, so they have a single circular piece of DNA, and they go through binary fission because they are prokaryotes. So if it's on that mitochondrial DNA, it's of course going to be found in the egg. Linked genes has to do with that it's on the same chromosome. So if you inherited this chromosome, then you of course will inherit all the genes that are on that chromosome. So the theory is that they are linked together because they're located close together. Can they be separated? Yes. If you go through crossing over, it can separate those linked genes, but the closer that they are to each other, the less likely they're going to get separated. So somebody says, are we going to go over some pedigrees? Yep, here we go. Let's look at a pedigree. So here you see that I've got a male and a female, okay, and then they have all offspring that are going to be not affected. They don't have that shaded. Shaded usually means affected. An open circle or square, it means that it's unaffected. The squares are our males and the circles are our females. So here you see we have all these offspring that are unaffected, okay? So here if I look at these two are unaffected. 1 second. Yeah, so these two are unaffected. And then as I noticed, there is an offspring here that is affected. So see how we've skipped a generation? It was in the grandparents. It was not in the parents, but it was in the grandchildren. Okay. Because of the fact that it skips the generation, that tells me that we're looking at some type of recessive trait. Okay. Now, because of the fact that this female is affected and she does not, and the male does not have it. Males have only one X chromosome. So if this was a sex-linked trait, this male would be affected. And so he would have had that X chromosome to be able to pass it on to our female. Right here, as we said, it's recessive. So she has to have 2 of those X chromosomes in order for her to show this trait. And so we have an unaffected father, which means that this can't be sex-linked. So this tells me that it is autosomal recessive. Will they keep the pedigrees whether males or females? I'm sorry, males or squares and circles or females, or do you memorize that? I believe the questions that the sex matters, I believe they've told you. But it's not really too hard to remember. But if that's something that you have trouble with, then I believe they do give it to you on the question. In terms of if it's autosomal or recessive and, the inheritance patterns of it, they might not tell you it. They might just show you the pedigree and then ask you what is the form of inheritance. So here we're looking at it. We can see that it is in every generation, okay? So we can see that we've got this female and these males, of course, and we do see that it is in every single one of them. How do I know if it's autosomal dominant versus sex-linked? Traditionally, an autosomal dominant one it's hard to differentiate them. The big thing to focus on them is looking at the X chromosome. And so here we can see that we've got the affected female and there was affected males. This is an unaffected male versus here I have affected male, affected female. So I see that this is X-linked recessive. So as I said before, in order to get that affected female, it had to be with the affected male and the affected female in order to pass that on. So they're going to make it obvious, whatever it is that they do. So here would be an autosomal dominant one. So the way that we know that is it's found in every generation. It's kind of equally found in males and females. The female, a little bit more in this diagram of it. But overall, we do see that it's going to be equally found. So autosomal is equally found in males and females. Dominant meaning that it's in every single generation. What's the difference between X-linked versus Y-linked? X-linked means that it's on the X chromosome. Y-linked means it's on the Y chromosome. Theoretically, the only way that you can get Y-linked is if you are a XY individual. There is, you can have crossing over, and crossing over can allow for some of the Y chromosomes to end up on the X chromosome. I believe the SRY gene can actually move over that way. But so you could have crossing over, but the logic of Y-linked is that it's going to be on the Y chromosome. And then looking at this one, we can see that, okay, if we have affected males, none of the offspring are affected. But if we have an affected female, look, all of the children are affected. Again, looking at affected female, all of the children are affected. This is screaming either mitochondrial inheritance or chloroplast inheritance, okay? Because of the fact that it is an affected female with all of her offspring. If all of the offspring are affected from that female, it screams that we have some type of mitochondrial inheritance or chloroplast. You will have none of the males being able to pass it on. They do not have the mitochondria. They do not have the chloroplast to be able to pass on. So looking at the chromosomal inheritance, we're going to have a lot of variation. First thing is crossing over that we saw during prophase 1, independent assortment that we saw in metaphase 1. The law of segregation, so we can see that these alleles are going to separate and be moving into opposite poles, which means they're going to end up in opposite cells. And then random fertilization, any sperm confused with any egg. And so that's where we see this random chance. I believe it's like 8.4 trillion combinations can be made when we look at all the different possibilities, just looking at crossing over. I don't think that takes into it takes independent assortment in, but I don't think it takes for crossing over in. So there's so many combinations. You are unique. So here you can see, of course, that crossing over. And then here you can see the law of segregation, the law of independent assortment. So here we're seeing that they're either lining up where we have paternal on one side and maternal on the other, or they can be vice versa. And then the law of segregation is just meaning that they're moving to opposite poles. Okay. So in terms of the chromosomal basis of inheritance, that means that there is some type of chromosome, something that gets passed from parents to offspring. And that's what is being, how we're inheriting our traits. There was a blending hypothesis where it was just that there was information being thrown down. It was all blended together, but really it's a chromosome. There's a genetic information. And then there are genetic disorders. They're probably not going to there's no specific genetic disorder you need to know. If they have you coming up with one, they're either going to tell you the genetic disorder, they're going to say there is a genetic disorder, and then they'll tell you how it's inherited. But so just giving you a little background on some things. Sickle cell disease is a autosomal recessive. It has to do with that it's a single allele. It's actually going to be a point mutation. It's a missense mutation, if I remember correctly, where we have one amino acid substituting for a different amino acid. You have Tay-Sachs. Tay-Sachs is going to be also another recessive trait. And then Huntington's disease is autosomal dominant. So we're going to find that it is going to be on it's a dominant allele, and it's due to duplication that takes place. And so you have too many of a certain gene, and that leads to Huntington's. Chromosomal changes, so you could have differences in our chromosomes. So here, this is called a karyotype. So you take, during metaphase 1, you kind of stop the process, and then you can actually look at all the chromosomes. They line them up based on their length and then their color pattern. So the reason why they're called chromosomes, they take up dyes, they take up colors. So we call them chromosomes. And so if we organize them, we can tell that this individual is an XX individual. So this is a female. I can also see that this individual has 3 sets of chromosome 21. And so this individual has something called Down syndrome or trisomy 21. So we're going to celebrate them on March 21st. So on Friday, you should be celebrating Down Syndrome Awareness Day because of the fact that it takes place on March 21st, because trisomy 21, they have 3 sets of the chromosome 21. I see a question. For autosomal recessive pedigree, we did, could you go over how to map the alleles correctly? I'll do it in just a second. We also have Klinefelter syndrome. Klinefelter has to do with that there's 2 X chromosomes and 1 Y. Because this Y chromosome, this individual is going to be a male, and they have that extra X chromosome. So somebody asked earlier this week about a cat, calico cats. So calico cats are going to be the color that they are due to bar bodies, and we inhibit, basically they You methylate one of the X chromosomes, it causes it to decrease and become like itty bitty and super condensed and becomes something called a Barr body. So our Klinefelter syndrome, these individuals have a Barr body. They only, they have one extra chromosome. So they can condense one of these into a Barr body. And though you can have a calico male, it just happens to be that they have, extra sets of X chromosome. And then Turner syndrome means that they have one X chromosome and then they don't have a second X chromosome. If mutations are the only source of genetic variation, then why does crossing over and random fertilization they don't create genetic variation. They're going to increase genetic variation. So the only way you can add new genetic variation to a population, like a group, has to do with a mutation. Mutation adds a new allele, a new variation to it. While crossing over, independent assortment, random fertilization, those are all going to reshuffle the existing alleles to give you new combinations. So crossing over can create a combination of two different traits that are now going to be inherited in one individual. So that creates variability and variation. Okay, so I think I answered that one. Happy birthday, Crystal. Okay, so environmental effects on phenotype. So we have this thing called phenotypic plasticity. Phenotypic plasticity has to do with that the environment can affect how the genes are expressed. So you can have the exact same genotype, but you can show slightly differences in your phenotype. So here, if I put an ice pack on the back of this rabbit, bunny, whatever you want to call it, it's going to end up growing a darker color on its back because of that colder environment causes it to kind of be darker colored, probably to absorb more of the light radiation to keep the body warmer. That's probably why its feet and its nose and its ears are warm or dark, due to the fact that those were probably colder areas for that for the expression of those genes. We also see the same thing in these birds. Based on their diets, they actually eat something different, and because of their diet, they look different. So this one's like yellow, and this one's kind of a red color because of the differences in the food that they have consumed. And then, of course, the normal example of hydrangeas. Hydrangeas, based on the pH of their environment, they're either going to be blue or they're going to be kind of pink. You've done 7, 8, 1, 2 so far. You cooked. You probably need to catch up a little bit. So on to the questions that some people ask. So what is a sex-linked mutation that can be passed down? Sex-linked mutations has to do with that it's on the X chromosome, so it can be passed down from one generation to the next. Males are more likely to get something that's sex-linked because they only have one X chromosome, while a female has 2 X chromosomes. And it said, would this be in the mitochondria of the mothers? So we're not going to a sex link doesn't have to do with mitochondria. Mitochondrial inheritance is going to follow the mother because of the fact that she's the only one that has the mitochondria to be able to pass it down. And then here's some examples of X-linked mutations. So hemophilia is an example. Red-green color blindness is an example of a sex-linked trait. Duchenne muscular dystrophy, and then fragile X syndrome. These are all examples of things that you would see on that X chromosome and are X-linked. As I said, they usually aren't going to tell you what the genetic disorder is because they don't want you to use your outside knowledge to answer whatever question it is. So they'll probably give you a pedigree, and then from that pedigree, you'll have to say if it's dominant or recessive as an autosomal or as a sex-linked, instead of them looking at you and saying, oh, not looking, but if instead of the question saying, this pedigree shows hemophilia, what type of inheritance is it? Well, if you already know hemophilia is sex-linked recessive, then you're not going to use that pedigree, and they're testing, do you know how to read the pedigree? So they're going to probably not tell you what the genetic disorder is. So here you can kind of see color blindness. So we see a half shaded circle. That means that it's a carrier. So this female is going to have one dominant, one recessive allele, while you notice that the male is going to not have the color blindness. And so this male right here got that colorblind allele, so he is affected, and then you have just carrier females. So you can kind of follow the path through it. I find that with pedigrees, I need to kind of figure out which one am I going with, and then I test it. So if I think that it's a dominant or recessive, I think whether it's autosomal or X-linked, whichever one I pick, I then test each of the individuals in there to make sure that it does follow that trend, so that I'm not, because they're going to make it obvious. The goal is not to trip you up. It's not going to be oh, I got you. You didn't look at this one little thing. Like, they're going to make it obvious when you have to figure it out. Can I understand between autosomal versus sex-linked traits? Autosomal means it is on one of the 22 chromosomes. So you have, here, we have 22 chromosomes that are autosomes. So all these are autosomes. And they're just cell, I mean, they're just chromosomes that make up the normal proteins of your cell, right? And then sex-linked has to do with it's on that X chromosome or that Y chromosome, okay? And so we find that it follows the sex of the individual, okay? But there are other genes that are on this, as we said, hemophilia, I think male pattern baldness is also a sex-linked trait. So like, I guess it wouldn't be male pattern baldness, but baldness is an example of a sex-linked trait. But again, we find it mostly in males because of the fact that they only have that one X chromosome. And so that's the difference really between autosomal versus sex-linked. Is autosomal just means it's on one of those 22 autosomes, and sex-linked means it's on one of the sex chromosomes. So we need another chi-square problem. So someone says, can we do chi-square? Yeah, of course we can do chi-square. Okay. So with chi-square, this is an example from 2003, so really long time ago. And we're just going to do the 1B. So they have the fruit flies, and the phenotype of eye color is on a certain locus. They're saying a capital E is the dominant allele, in lowercase e is a recessive. They cross a male fruit fly and a female fruit fly. The male is going to be wild type and the female is white-eyed. And they gave us a 1-to-1 ratio between the wild type females and white-eyed males. And so that screams out to me that it must be some type of sex-linked, because of the fact that why would the males all have one trait and the females all have another trait? So that screams that it's some type of sex-linked trait. And so I'm going to make the assumption that when we move into our question, which is asking about a chi-square to analyze my prediction of my genotypes, I'm going to predict that the female is going to be heterozygous, where she'll have one dominant, one recessive X allele, and then the white-eyed male is going to have the recessive X, and then, of course, the Y chromosome. So when we do this cross, we first, who is it? There's the Chi-5 by Jay Daly. She came up with this last year, and I use it in my PowerPoints. So first thing, null hypothesis. What is our null? The null is that the parent genotype is going to be this. As I said, heterozygous female, homozygous recessive, or hemizygous male. Okay, where she's got dominant recessive, he's got just the recessive. If we do that cross, I see that 1/4 of each of them. And look, I kind of see that I do have that 1/4 between all of them in that cross. Okay, and so I observed there being 23, 31, 22, and 24 of my different ones. And since from my Punnett square, I see there's a 1/4 chance of each of them. So 100 flies divided by 4 gives me 25 of each of them. So I have 25 that I would expect for them all. Okay. Now, I always use this chart when I'm doing my chi-square. So that would be my observed, my expected, my observed minus my expected, my observed minus expected squared, and then the observed minus expected squared over expected. So basically take this one minus that one. So 23 minus 25 is going to give us a -2, okay? And then I take this value and I square it. So -2 squared gives me 4. And then I'm going to take this value and divide by my expected. So 4 divided by 25. And I would do that for each of the different options. And then I'll add them together, and that gives me my chi-square of 2.0. Someone says, if they ask us to run a chi-square, where do we write the null hypothesis? Next to calculation, do we need to identify it? I don't believe they give us a table. So sometimes they give you a table. This question, they gave you a table, but all they gave you was observed, expected, and then the final answer for it. So you had to go through all the middle points yourself. And so actually, that wasn't this question. That was the fruit fly question about the choice chambers. That was in 2013. Anyways, so sometimes they give you a chart, sometimes they don't. I always draw the chart anyways. So what you'll see is you're either going to have a spot to write the null hypothesis, or when you give your final analysis, you're going to say, I failed to reject my null hypothesis, I rejected my null hypothesis, and then you can state what your null hypothesis is if it's a free response question. If it's a multiple choice, you don't need to tell them you're null. So how do I determine what to do with this? Well, there are 4 different classes here. I've got, wild type male, wild type female, white-eyed male, white-eyed female, right? I got 4 different options. So my degree of freedom is going to be 4, the number I have, minus 1, which would be 3. So I'm going to look at degrees of freedom 3 and my p-value of 0.05. That just means that I'm 95% confident. You can use the 0.01. That just means that you're 99% confident of your answer. So traditionally we use the 0.05. If they want you to use something different, they traditionally will tell you in the question. But whenever you're doing a free response question and you're going to state it back to them, you want to tell them what degree of freedom you gave, and then what p-value you used, and then what the critical value was, and you're going to compare your value to that critical value. So I'm going to find that my value of 2 is less than the critical value of 7.81 at a degree of freedom of 3 and a p-value of 0.05. This means that I'm going to fail to reject my null hypothesis. The data closely matches my expected. Okay, the numbers are small and they're very close together, which means that I can't reject my null hypothesis. Okay, so we fail to reject. We don't really accept the null because you can never accept something as true, but you can fail to prove it wrong. And that's what we're doing. We're failing to reject that null hypothesis. I don't know if I can go over unit 4. I can't go over that one. That is a secure question, number 1 and number 2. I don't have that question memorized. So someone says, can you help me understand karyotypes? How do you analyze them? How do you go over them, please? And then they wrote me much love. Aw, how sweet. So here you can see a male karyotype. We're going to have 2 of every single one of our chromosomes. Okay, so you just put them in order from longest to shortest, and then our sex chromosomes go last. And so you'll notice that they have 2 of every single chromosome. So this would be a male, and they're a normal male because they do not have any genetic abnormalities. And then same thing here, I have my normal female. There's no genetic abnormalities. They have 2 of every single chromosome, and they have 2 of the X chromosomes. So that's where we notice if it's a male or a female is those XY or XX. If we're looking at other things, there was a question a couple of years ago about zebrafish, and they use a ZW system. So just use your question. It'll usually tell you what kind of sex chromosomes they have. They'll also tell you the inheritance of it. Like, so for the example of that zebrafish question, there was ZZ was actually the male and ZW was the female. So there were slightly differences in what we're used to. And so looking at this, we can kind of figure out, well, what would be the genetic disorder from this? So I can see that this individual has 3 sets of chromosome 21, which we talked about earlier, means that they are going to have Down syndrome. And then this one, we can see that they have XYY. So they have 2 of the Y chromosomes. This is due to non-disjunction that took place during meiosis. So if those homologous chromosomes do not segregate, if they don't move apart, then we end up with an additional set of chromosomes or a different additional chromosome of a certain one. And that's what leads to that. And so then, do you have any questions? No more questions yet. So explain the use of the probability equation. So here is our probability equation. They said if A and B are mutually exclusive, then PA or B is equal to PA plus PB. What? And then if A and B are independent, then probability of A and B is P of A times P of B. Okay. So the best way to explain this is to use an example. Okay, so here we see our Punnett squares that we did earlier in the lesson, right? So we have 2 heterozygotes, and in here I told you that 3/4 of them were yellow. Well, how do I know that 3/4 of them are yellow? Well, there's 1/4 right here that's going to be homozygous dominant that makes them yellow. And then there's 1/2 or 2/4 that are going to be heterozygous that are also yellow. So the probability of homozygous dominant plus the probability of heterozygous gives me the probability of the yellow allele. So looking at the single Punnett square, one single Punnett square, that's going to be where we're going to do the addition rule. We're going to add them together. So P of A plus P of B, right? That's how we would use the first equation. Now the second one was when we were multiplying them together. So we were multiplying our probability of yellow and the probability of round, and that gave me the probability of yellow and round, which is my second one. They're independent. They're on 2 different Punnett squares. And so that's when we're going to multiply. And so with the law of the probability, the first one's talking about looking at the same Punnett square and adding them together. And the second equation is looking at multiplication rule. Okay, so that is all that I have in terms of pre-submitted questions. So please, if you have any additional questions about Unit 5, please throw them into the chat. I'll probably kill a little bit of time just because of the fact that I know that there's a little bit of a lag between you and me. Why does having extra X or Y chromosome have less of an impact on autosomal chromosomes? I don't understand your question, because if you have an extra X chromosome, there is a genetic abnormality. Same thing as if you have an extra Y. You're having too much of the genetic information, and the body actually can't take additional amounts of those chromosomes. And that's what's happening with most of these genetic disorders, is that they have too much of a certain chromosome. They have, chromosome 21 leads to Down syndrome because of the fact that they have too much of the genes that are on chromosome 21. While I kill time, let me just remind you. Here is the website. I've got, tons of resources here for you. So last year's crew, 95%, got a 3 or better on the exam. And when I asked them what were their most helpful tools, they mentioned the unit review videos, the TikTok videos, and then the review games. And then I've also got links to like everything in here. So here's that 396-page review guide. I broke it down by I can statements and then the topic questions. Then I've got every multiple choice question, every free response question that's been released. I've gotten it posted in there. So it's easy for you to be able to, practice on certain FRQ questions or certain multiple choice questions. Here's the 120 review games. So all you gotta do is click there and it gives you the code that will take you to the quizzes game. Here's all those FRQ videos. Here's resources that were made by like your peers. So sometimes near peer helps out. One pagers. These are one pagers where I just took the PowerPoint and put them into like a single page. So you could use that to study. Here's the results from the past year. So if you're wondering like, oh, I want to make a 5. What resources do I need in order to make the 5? So here is the is the quiz, not the quiz, the form that I submitted to the students. And I said, hey, what was your score and how did you do? And what did you use to prepare for the exam? That's all the information that they gave about, what they would use. And so all of that is available for you. And you they're all still here. You can drop down, go to everything you need there. Okay. And I've got all the stuff. There's a study plan here. So let's assume you're not watching this video till, I don't know, 4 weeks for the exam. I do have a 4-week review guide there for you, as well as I have a 10-day countdown to try to help you to review last minute. Okay, so not sure how to word it, but how do you apply the probability to chi-square problems? I know how to do chi-squares, but I'm unsure how to apply the ratios to it. So when we did our ratio here, we looked at the ratio, right? There was a 1/4 chance. So here's our ratio from our probability, our Punnett square. And we said, okay, well, if 1/4 of them I expect to be female, then 100 times 1/4 gives me 25. And I did that same math for each of these. It just happened that they were all 1/4. So I was able to just say, 100 divided by 4, and I made them all the same. But if your question ends up being 3/4 of one and 1/4 of the other, you would just do 100 times 3/4, giving you the 75, and then 100 times the 1/4, giving you the 25. So it all depends on what you expect from that problem. Is it best to study by using notes or just watching the videos or doing the games? So everyone has different ways that they study. I can't tell you what the best way to do it. My personal opinion is that the more you interact with the content, the better it is. And the more active you are in your learning, the better it is. So sitting here and just watching someone's videos aren't going to be the most helpful tools for you. Now, if you went and paused it every couple minutes and you jotted down summarizing notes, that's a more active process. If you created questions, that's an active process. So doing something to kind of help keep you motivated, pausing and taking your own notes, downloading the PowerPoint in advance and annotating the notes that are on there based on what the person in the video is saying. There's multiple creators out there. All you have to do is just say AP Bio Unit blah, and you're going to find people. There's Hey Now Science. There's, Absolute Recap. There's Bozeman Bio. There's Carrera. There's Gabe Poser. There's so many people out there that are making videos. And after we went through COVID, there's a lot of teachers who just posted chapter videos. So you've got tons of videos there. In terms of if you use the review games, going through the games and seeing what you get wrong. If you got it wrong once, there's a chance that there's a misunderstanding. So looking through the things that you're getting wrong and figuring out, okay, did I not understand the information? Did I not understand the question? Or did I just rush through this question? So figuring out where are your hangups, okay? And remember, you don't have to be perfect on the exam. I think you only need probably a 60% in order to pass. And that is probably me just being a little harsh, but still, you probably need an 80%, for your 4. You probably need 85, 90 for the 5, maybe more 75, 80, but for the 4. But, there's these cut scores and there's no way for us to know what those cut scores are. My big push is that you're going to do the best you can. You're going to leave it all in the test. You're not just going to, whatever you did on the exam was your best. You did everything you possibly could to prepare yourself for the exam. So there's no way that you're oh, well, I don't care about it. You're sitting in a review session right now and it's currently 30 minutes in, 42 minutes in. You want to do well and you're going to do well because you are putting in the time and the effort to do well. Can I explain the term multiple genes? Multiple genes just means that there's, multiple genes for a certain allele, not allele. There's multiple alleles for a certain gene. So blood type. There's the A allele, the B allele, and then there's a recessive allele. And so that would be multiple genes. We've got 3 sets of those alleles. Okay, no, 70 to 75 is not a 5. That is like a 3, like a 3 or a 4. Like I said, an 85% for a 5. 85% for 5. Like maybe 80 to 85. I don't know. I would go 85%, honestly. I would go 85 to 90% if I was to guess. I would probably go like 75 for a 4. I really don't know, though. Like, honestly, these are just me throwing out numbers. Okay. Is there one unit that is harder than the others you study more? Every student has a different opinion on what the hardest units are. My students have always told me that unit 3 is hard, unit 6 is hard, and then unit 7 is hard to them. I personally think unit 8 is hard because I don't like ecology, and thus, since I don't like it, I have a harder time learning it. So if you're in the same mentality where you find that something's hard because you just don't like it, find something about it that interests you, find something about it that's cool, and that'll help you to kind of get over that hump of the difficulty. The more you're interested in something, no matter how hard it is, if you're interested in something, it doesn't feel hard. It just feels like, work. You're doing your best, and that's, again, all that matters. Okay, so are there any other questions before I log off? I think I've said my gist. So remember, AP Bio Penguins, just success. Y'all are going to rock this exam on Cinco de Bio.